Awesome. Then it's a pleasure to introduce today Dr. Jula Naso, who is a pathologist, anatomic pathologist at VGH. Julia is a clinician scientist. She did her MD PhD at UBC, her PhD part with Marco Mara. Um, she's done some seminal work in genomics in particular, and more recently in pulmonary pathology. She did a fellowship training in pulmonary pathology at the Mayo Clinic. Um, and as a clinician scientist, her interests include translating novel molecular testing into the clinical realm. So thank you for agreeing to present today, Julia, and please go ahead.
Oh, my pleasure, thanks. And I'm always grateful to you for all the support of the department here in enabling me to do this work while also being a pathologist, and for all the mentors that I see online today. You all see the slides, I assume. You hear me okay? Excellent. My fellowship is in lung pathology, so this is approached a little bit from a lung viewpoint, but hopefully you'll see how it relates to squamous cell carcinomas at other body sites too, and maybe it'll include your favorite one. To step way back in the context of cancer pathology, lung is a heavy hitter here with almost a quarter of our cancer-related deaths from lung cancer. And within that, I can pencil in, there's a subtype of lung squamous cell carcinoma. It's the second most common type of non-small cell lung carcinoma, which is squamous cell carcinoma. And that would be about probably a quarter or so of lung cancer deaths. It's about as lethal as other non-small cell carcinomas, and about a quarter to a third of our non-small cell carcinomas. So that's a fairly significant piece of the pie. But I find it curious that a lot of times we group it into this notion of non-small cell lung carcinoma. And I think I'm not alone in thinking that maybe we should stop lumping these all together, because squamous cell carcinomas are probably very different biologies than adenocarcinomas. They have different genomics, different response to treatments, different treatment options. So to me, at least, it makes sense to give them their own piece of the pie, to study them separately. And I approached this starting to think about what could I do that's a problem that's more specific to squamous cell carcinomas than other things that might need some unique solutions. The other kind of motivation for thinking about squamous cell carcinomas is, in a way, they seem to me a bit like this unhappy middle child, the forgotten middle child, in that I find in lung cancer research, people seem to want to study either things that are very aggressive, like small cell carcinomas, or things that affect never smokers, like adenocarcinomas, or things that have cool targetable driver mutations, which again is adenocarcinomas. And squams are kind of this in-between zone. They're not the most common, not the most rare, not the most aggressive, but I think they are still a significant portion of cancer mortality, and certainly worth a look. So in a clinical pathology practice, what I was finding was one of the biggest concerns is just figuring out which squamous carcinomas are from where and how they're related to each other. There was three main questions. We can break it down into, if you see something squamous, it's sometimes very tricky to figure out where it actually came from. If it's like in the brain or in the liver or some soft tissue site, where did it actually start? Another conundrum is if it's in the lung, and the patient also has squamous cell carcinoma somewhere else that could generate a squamous cell carcinoma. Is that thing in the lung metastatic from their other site, or is it a new primary cancer? Because all of those sites could generate their own. And then finally, in the lung itself, if you have multiple squamous cell carcinomas, how do you know if they're intrapulmonary metastasis, which could be synchronous, identified at the same time, making it a T3, or separate things, which could be a T1A even for each of them? Or if they're metachronous, like, is one of those a recurrence or a metastasis from the earlier one, or is it a brand new disease? And I think some of these questions can really influence how it's approached for management. If you've got widely metastatic disease, you probably don't want surgery, whereas you might want some kind of systemic treatment. And that difference between surgery versus systemic therapy is a major one for patients to face. So how do we address these questions, and what are the barriers to addressing these questions that result in them not having been addressed yet? We like morphology, of course, but morphology is tricky for squames in that a lot of it can overlap between these sites. You can't just look at it and know where it's from, nor can immunohistochemistry tell you where it's from. We don't already sequence squamous cell carcinomas in the lung. In fact, you have to have an adenocarcinoma component in order to be eligible for a local focus panel testing. So there's no pre-existing or already done sequencing data for these tumors. Kind of an opportunity to come and create our own, but there's certainly nothing we can leverage pre-existing. There's many places they can come from. These are all very prevalent things. They often have similar risk factors, so people can be reasonably likely to have more than one tumor develop of squamous histology. They have pretty overlapping driver genes. You can't just look at which gene is mutated or which gene is amplified. And when these have been studied in the past, there's often a pretty limited sample compared in the sense that people will pick maybe their two favorite types of squamous cell carcinomas and see if they can tell them apart. But there's certainly a lot more than two body sites that can develop squamous cell carcinomas. How do we take into account that diversity of possible origins? This is a very busy plot, but I think the overall message here is if you compare squamous cell carcinomas of different origins, they have a lot in common and some differences, not quite enough differences that you can tell them apart, but enough differences that I think it is reasonable to also study them separately, so that just because we know some things about head and neck squamous cell carcinoma doesn't necessarily mean that it's true for lung squamous cell carcinoma. And that further motivates me to have a look at the lung squames. In the past, in terms of efforts to try and figure out the site of origin of squamous cell carcinomas, I think the greatest success has been obtained from methylation-based classifiers. That might be my bias, take that with a grain of salt. But I wanted to show you here one example of an approach to primary of unknown origin tumors using methylation. This is from a prior publication. You can access it online. You can upload your own data sets and it'll classify it for you. And it'll put it in one of these lovely little pictorial groups. But you'll notice the squamous cell layer is a little bit minimal. There's only 3 categories. They lump together head and neck and esophageal. Cervical, I mean, you might even be able to figure that out on your own with HPV testing. So I think we have some room to grow that branch of our tree by looking specifically at squamous cell carcinomas. Because often as pathologists, you know that you can probably figure out if it's squamous. It's just after that, trying to figure out how to localize that further. Okay, so this brings us to the first bit of our research, which I credit hugely. Alan developed this classifier and he put together the figures for this. So massive credit to him. And you'll note that there is a large team behind this that I have to send huge thank yous to. A lot of the biobanks locally pitched in for this. So you'll see some prostate folks, some head and neck folks, some ovarian cancer folks, and many of my other mentors, some folks from Toronto who have also done work in this area. So thank you to this crew. This manuscript submitted now and just waiting for a review. It started by looking at pre-existing data from TCGA, and there's almost 1,500 cases of squamous cell carcinoma that have methylation data. This is from methylation arrays. And if you just take all of the methylation data and you cluster them and figure out what is more similar to what else, it's interesting that tumor tends to cluster separately from normal. But we found it very promising that this top bar, the site of origin, it more or less already shows some clustering, even without trying to pick out genes to help it cluster. Just intrinsically throughout the signal, there was some clustering. We also thought it was helpful that the primaries and the metastatics, tended to be rather dispersed. So even when something becomes metastatic, it probably retains some signal of where it came from. Again, giving us hope that this method would work. Less fortunate is that the HPV ones tended to cluster a little bit separately. So we're thinking that that might be a bit of a challenge in our algorithms to try to identify these HPV related ones and break those down into the different sites that can have HPV associated tumors. So training this algorithm, and again, Alan did a fantastic job developing a neural network that took into account all of this methylation data. If we used samples that were from the same sources like TCGA, other online data sets, but not exposed to the training of the algorithm, so held out in that sense, we can get a fantastic accuracy, about 96%, AUC of 0.99. This is like predicted versus true. Almost everything lines up right in the middle. Then if we take an external test set, so this is a mixture of some local cases and cases from Toronto, a few other data sets from the literature that weren't in the training, we maintain a pretty high accuracy, better even than our internal cases. So we think that it shows pretty robust classification despite batch effects of where these samples are coming from. There's kind of a recurring theme that HPV negative cervical cancer never classifies the way I want it to, but for the most part, other things will classify well. This makes AUCs that are a little bit goofy looking because they're actually so good that they look a little bit goofy. And that was very encouraging to go on ahead with this. The other thing we wanted to think about sooner than later as well was making a threshold for these predictions. Every time this network is run, it gives you a score for each of the 5 possibilities. And we were classifying things as, well, the option that had the highest score is the one that we'll call it. But what if we put in some kind of a threshold that said, your highest call has to be above this line in order to be called anything? Can that make your accuracy even better? And it does. It pushes it up from the 96 or 97 to about 99% accuracy for those who are particularly perfectionistic. And then it would leave about 12% of samples unclassified. So you can get a good sense of whether you should really truly believe this result or not. This plot is a little bit tricky, but the green ones are accurate predictions. The red ones are not accurate. And so you can see that the correct predictions do tend to be at the very high confidence end of our spectrum. All right. Then we went on to ask, so far this was on primary tumors, which, I mean, in practice aren't the ones you really want to know about. You want to know about metastatic site tumors. So the question then was, can we still tell where it came from, even if it's in a background of tissue that's different than the tumor itself? We were a little bit worried that the background tissue might contaminate our data and like supersede the results from the tumor itself. But sure enough, even at metastatic sites, we still got our 96% accuracy for predicting where this tumor came from. And perhaps the most challenging metastatic site to predict in theory would be lung, because that's one of the options for what to call this tumor. And we were encouraged that 34 out of 36 of our cases that were metastatic to the lung were still called as something other than lung, which they truly were. It wasn't just predicting lung based on the background lung tissue. All right, and then we had a look as well at tumor purity. Maybe these incorrect calls were just because they were really low tumor content samples. We only tested down to about 40% tumor content, and that one worked. There's not a whole lot in this lower range, but it looks like it holds up pretty well regardless of tumor purity, at least in this kind of higher half of the spectrum. It's probably other things then that are causing the few miscalls. And exactly why is something still to be determined. We didn't notice any correlation with treatment or any other variables. So TBD as to why these make a few misses. We did notice that the esophageal ones were ever slightly more often misclassified, like in like kind of a 93, 94% range as opposed to 96%. But very encouraging, I think, to move forward. The next thing we asked is, can we see inside this black box of why is it calling the sites that it's predicting? And for that, we looked at which genes and which sort of pathways were being amongst those that would be affected by the changes in methylation site that fed into the predictions. And we noticed that homeobox genes were among that, which kind of makes a lot of sense because those are genes that are involved in patterning what tissues develop where. I put up the plots with the individual genes if you see any that you really like. I noticed GATA3 is in here, actually. It's kind of to do with bladder as well, which I thought was kind of cool. But I think the strength of this approach is that there are so many different areas of the genome that feed into the methylation sites that are part of this algorithm. So even if you don't cover them all in your particular assay, if you cover a lot of them, maybe you can still make a pretty accurate call. The problem with this, though, is that if it's anatomical patterning genes that are driving this classifier, there's a possibility that normal tissue could screw up your results. So if you tested this classifier on samples of purely normal tissue, 92% of them would still be correctly identified as the site they came from, meaning if there's a whole lot of background in your sample and it is one of the options for the classifier, it's possible it could be miscalled. I already showed you the data for vets to the lung being still accurately called, but it's possible that if those were lower tumor content samples, they might start being miscalled as lung. All right, now this all sounds great, but the problem is if you want to do this in your clinical samples, how do you get methylation data? So I wanted to talk for a little while about some of the options. So far, the data that you've seen is from methylation arrays, which have some advantages. One of them is they're not super expensive. They don't need a whole ton of DNA. About 300 nanograms is probably fine. We used 800 for our samples. They take a couple weeks. The caveats, though, I think the biggest one is that an array chip has 8 samples. So if you want to do this in a cost-effective manner, you should probably fill up all 8 samples on the chip. But the odds of getting 8 patients that all have this clinical question of squamous cell carcinoma of unknown origin at the same time is pretty low. It would be really nice if we could do 1 sample at a time and not have that make it 8 times the cost. So far in Canada as well, there's 1 lab, I believe, that's clinically accredited to do this. There's many other excellent research labs, but the clinical accreditation, if you want to call this a clinical test, would be a factor to consider. From speaking with our neuropathology colleagues, it sounds like send out for a clinical methylation array is pretty frequent, like every couple of weeks. You can correct me if any are on the call, but it sounds like every couple of weeks or so, and it takes about 2 or 3 weeks. So definitely doable, but not necessarily every day. And that is for the question of brain tumor classification as opposed to site of origin. I've yet to hear from any pathologists who've done a clinical site of origin test, but I'd be very curious to hear if you have done any. The other caveat of arrays is that they only have just under 1 million or so methylation sites covered in the latest versions of the chip. They don't give you any other data other than methylation. But hey, at least they work on FFPE. And that is a big plus I shouldn't undersell, is being able to work on FFPE tissue. The option, which you've probably been staring at this whole time, that's an alternate, is this Oxford nanopore sequencing. I put up a picture of this very cool little sequencer. Fits in like the palm of your hand in its smallest version. There's bigger versions too, and GSC has a bigger version. This one belongs to Dr. Yip. Thank you for letting us photograph it. But nanopore sequencing is very different in the sense that it is actually reading the nucleotide sequence of the DNA by the molecule traveling through a pore and looking at changes in electric current as it does so. And that can inform on the molecule passing through the pore. In doing so, it can detect modifications on the bases that are there. So it can detect methylation on that cytosine or other possible modifications on your bases. That way you don't have to bisulfite treat it, you just put it through the hole. Caveat is it has to go through the hole, which means that if you're formalin fixed and cross-linked, not going to go through the hole, you'll plug those up, and it's probably impossible to make work on FFPE tissue. So you need fresh frozen. And I should have put, or you could probably do this on, not probably, but people have done this on circulating tumor DNA. So also feasible for plasma samples. Good news is it doesn't need batching. It still actually takes a pretty low amount of DNA because you don't bisulfite treat, like you don't degrade up 80% of your DNA as you otherwise would if you bisulfite treated. If you batch them for research, it's certainly a reasonable cost, and you get the whole genome. You can try and use some software tricks to bias towards covering the regions you want, if you want, but there's certainly an option to get a whole genome from this. And you can also kind of infer copy number too, based on how many times you sequence different regions in your specimen. All right, so as you might guess, we're going to talk a little bit more about using nanopore sequencing for this. There are certainly some groups who have already done this in the context of brain tumors for the brain tumor classification. As some of you will know, the WHO now for neuropathology includes methylation as some of the diagnostic criteria. So it's become integrated in their practice faster, I think, than in many other areas of pathology. And they're really leading the way towards integrating this. These other studies I've listed, their focus was on, could we even do this fast enough to do it intraoperatively, which is pretty cool. The study was speaking about how it, in like 90 minutes, you can get enough CPGs that you can be sort of accurate as to your brain tumor classification. Only like 15 minutes of that was sequencing time, and the rest was sample prep, I assume. So we probably don't need site of origin classification intraoperatively, but it would still be kind of handy to be able to get in a couple days. It could guide what other investigations a person might go under to try and figure out more evidence as to what their site of origin is, or could help them know sooner whether they need to plan for chemotherapy or surgical resection. This can be really handy if they're out of town and trying to figure out where to live. So I think some faster results certainly has its advantages. And certainly faster if you take into account that you could do 1 sample at a time instead of waiting for 8. Makes a very big difference in your turnaround time. That said, if you're sending everything to Toronto, you can batch with the rest of the country. So it's maybe not as long to get 8 as you might otherwise think. Okay, So we were able to access some nanopore sequencing data to look at methylation in it through the help of the POG team. Huge thank yous to them. They've done this nanopore long-read sequencing on 16 of their samples that we show here. And they were all correctly classified in our classifier, except for one that was this case down here, and a lower confidence score as well. So you might notice that and not trust the result as much either. There was one that like barely got classified as the correct one, but it was a very strange scenario where this person had quite a few prior malignancies and had some very unusual genomic signatures for the thing that the clinicians concluded it probably is. So I think no one's quite entirely sure as to what the ground truth is on that one. One thing that we did was bioinformatically downsample the very deep sequencing the POG team did to see if we just did a really shallow sequencing of these cases on the nanopore, like down to the order of like 1x coverage, which would be covering each base in the genome on average just once. Can we still retain accurate prediction? Which sounds a little bit dubious if you start thinking about that means for each methylation site, you probably either don't cover it at all or maybe cover it like once or twice. The good news is that the accuracy, that's the red line, and the AUC, the blue line, still remain quite high, down to even like about between 0.5 and 1x coverage. They get a bit worse. But to be conservative, if you did about 2x coverage, you'd retain pretty much all of your accuracy. So this we found very encouraging, that we could get away with pretty minimal sequencing on these samples and still be able to run our classifier, which makes a big difference for cost considerations. The less sequencing you do, the more you can pool in a chip, and then there's a lower cost per sample. So we tried doing shallow sequencing on frozen samples from biobanks. This is to show that instead of just bioinformatically downsampling the reads, if you actually only sequenced it this much, could you still get good results? And out of 20 samples, we got 90 correctly classified. If you use a 0.8 threshold, then everything that passes that is correct, though 4 would be unclassifiable. So there is a way to kind of dissect out this 90% to know which ones to trust and which not. The only ones that didn't classify well were an HPV negative cervical cancer and a sample with very low tumor content. So I think if I'd been more stringent on samples for here, we might even have that. All right. And then the very last thing we looked at in terms of using this in practice, in the real world, you don't just have 5 or 6 types of squamous cell carcinomas. It's possible that there could be from some other body site, many of which are listed here, as the true option for what your unknown squamous cell carcinoma is. So we asked ourselves, if someone put into this classifier something that's not one of the 5 trained types, how would it miscall it? Would it be confident, unconfident, and what would it call it? So if we use that 0.8 threshold, almost everything gets called as something. There was a minority that were unclassified. And it was interesting that the way I got called is things that were HPV associated got called as cervical, probably because in the training, most of the cervicals were HPV associated. And then other stuff got called head and neck. As a lung pathologist, I really like this because it means if you throw anything in, if it's lung, it'll probably recognize it. And if it's not lung, it'll probably call it something not lung. You might not know what, but at least it'll tell you it's probably metastatic to that site. So I think that there is some utility there, even in the context of untrained types, just for figuring out lung versus not lung. We did try increasing our threshold, and that did not make everything unclassified. So I think regardless of what threshold you use, there is still a risk that untrained things will get misclassified as something in our classifier. All right, so just to wrap up that little piece of it, testing on internal and external cohorts, on tumors at metastatic sites, on nanopore sequencing tumors. We have an excellent ROC for this methylation classifier for at least these 5 types of squamous cell carcinomas, with maybe some options to use it for at least lung versus not lung if there's other options in your clinical differential. Okay, drawbacks. There always is a caveat. Some of the caveats here are to do with tumor content that I've mentioned, that we didn't test it on the lower range. So abundance of caution if things are lower than about 45% tumor content. There is more work needed to sort out the effect of treatment on these samples. So far, we didn't see any association with prior treatment with misclassification, but there's a pretty small number of pre-treated samples. I mentioned esophageal doesn't work quite as well. Untrained tumors will get classified as something. So bear in mind, there might be something other than the trained types as the true site of origin. And HPV negative cervical cancers are a problem. There was very few of those in the training set, so I'm not surprised that that's beyond its classification. So altogether, we have this methylation-based site of origin classifier for things that are histologically squamous cell that perhaps could help with finding what site of origin it came from. This should be accessible soon on the MAPCO website. We'll have a link to it in the manuscript when it gets published. And we gave it a very, very cheesy name. It was the best we could come up with. It's the Squamous classifier. So if you're thinking of a squamous cell carcinoma, it's the Squamous classifier. All right. Now, what if you can't get methylation data? We've talked about some options, but they're all a little bit tricky to get. And then the other scenario where this approach really won't work on is what if both of the two tumors in questions arise from the same organ system? Like they're both lung tumors and you're trying to figure out if your two lung tumors are related or not. That is where the second half of this comes in. Jen, Brandon, Samir, they all, I should have bolded Samir as well, apologies. But these individuals all really helped with the analysis and presentation of this. Jen presented this at USCAP and made these lovely slides. If the slides suddenly look prettier, it's because Jen made them. Thank you, Jen. And I'll go through some of the findings from this portion of the project. As you can guess, we're shifting gears a bit now to look at some whole genome copy number profiling, but still in squamous cell carcinomas and still dealing with this question of multiple tumors. All right, so a bit more background. If you think about how in the genomes of squamous cell carcinomas, there are a lot of CNVs. They've been described as just copy number chaos throughout their genomes. And that each of these different CNVs has a slightly distinct start and end point, just stochastically. They might span a common driver gene that's being selected for, but exactly where it starts and ends could be different. And that metastases should inherit some of the driver CNVs that arose earlier in tumor development. If all of these things are true, then you might think, well, between clonally related tumors, like a primary met pair, then there's perhaps enough conservation of CNVs that you can tell that they're related, whereas separate primary tumors might have different enough CNVs that you can tell that those are separate. The trick is trying to figure out where that cutoff is of how much overlap indicates something clonal versus how much difference shows something that's separate primaries. And how do we figure out that threshold? And sort of a even more superficial to that is how do we even get the CNV data from these cases? Is there kind of a clinically feasible workflow that can generate CNV data? Just to illustrate some of those ideas, Jen made this lovely figure of distribution of copy numbers across genomes in squamous cell carcinomas. You can see that there's lots, they're all over, they're shared by many of the different types of squamous cell carcinomas. So there should be a lot of copy number variations to put as input into this type of analysis. And on the bottom here is a plot from a published work by Amelia Lim, who's now local at UBC as well, if you know her. And in her study, she separated clonal copy number changes, which are the line, versus subclonal ones, which are shaded in yellow. And you can see that at certain points in the genome, there's a lot of clonal stuff and not a lot of subclonal stuff. So if you want to find a region that's likely to be a conserved change as its tumor grows, then there might be some options to pick on these areas that tend to get clonal alterations. All right, now this idea of using shallow whole genome sequencing to look at copy number is also not original. Huge thanks to Dr. Huntsman for introducing me to this idea. There's a paper from his lab on the top here where they were doing this on endometrial cancers and producing some lovely copy number plots. There's also a group in Toronto who's helped us with the mechanics of this using shallow sequencing, in part for copy number, but also in part for mismatch repair deficiency in their study, which is a kind of unrelated but also very cool application of shallow sequencing. There's some ideas as well that you could get homologous recombination deficiency signals out of shallow whole genome sequencing as well that Dr. Huntsman's group explored. So all sort of food for thought in terms of thinking about new assays for our labs. This one at the bottom I wanted to mention because they really highlight how low you can go for inputs, that if you have a tiny biopsy specimen, there's a reasonable chance you'll still get 25 nanograms of DNA out of that. You only really need 1 to 2 times coverage, and deletions as small as 5 kb you could probably detect. If you are at all thinking about doing shallow sequencing, this is a bit more detail on the method. For us, we used 0.4 to 1x coverage. Depends a bit on whether you actually use both paired reads or not. The paired end doesn't really add a whole lot when your reads are only 150 base pairs long anyway. But we use paired end sequencing really just because that's the most common workflow. So it's a bit simpler to get data faster if you use the same sequencing workflow as most other people do. This was an FFPE. In our hands, we went down to 100 nanograms and it looked really indistinguishable from using 500 nanograms for input. So I'm optimistic that it could even work for lower inputs too. The cost of sequencing now is kind of mind-blowingly not expensive. This $150 per sample, I think of that cost, only about $10 was the actual sequencing cost, $10, whereas everything else was library construction and DNA extraction. So if you are thinking about an assay that you might have to do twice for a patient, like once on each tumor to compare to, having each assay being not that pricey is a plus. For comparison, a lot of panel sequencing will be like $500 or $600 at least per panel that you run. All right, caveats is that you do need both the tumors if you're going to compare them. So not always do you have an available specimen of both tumors. And if you show they're related by this kind of a method, you don't know which one came first. So you don't know which of the two was the primary. That might not be an issue, but it's potentially less informative in that sense than methylation. It's a little bit sensitive to the quality of DNA. We did have some issues with if you do a robotic platform extraction, the DNA tends to be a bit more degraded and gives you very messy plots compared to a higher quality manual extraction. And to be cognizant of the size of deletion that could be detected by this. I'll show you on the next slide some methods of power calculation. And finally, we picked cases that had reasonable tumor content for this. All right, so if you ever think about running this and you want to figure out the power for how small of a CMV could you detect with given depth of sequencing, someone else has already thought of that. There's a website you can go to. It's here from a prior publication. You can put in what you know about your tumor and what power you want, and it can give you this curve of how many reads you need to get in order to, and at what tumor content you would get in order to get your indicated power. Long story short, about 50 kb is about the power you get for these metrics, which is pretty good considering that for fish, for comparison, for instance, a lot of fish probes will only have a resolution of maybe 200 to 300. Or sorry, 100 to 200 atoms, 100 to 200 or so for fish. So it's about at the level of fish resolution, perhaps smaller depending on your probes and your depth. All right, so to test this out, I'll show you some data today that's on an early batch that we did that is a pretty limited sample size. We have another 100 or so coming off the sequencer, hopefully any day now. But this we got a little bit earlier to have a look at. And there's 3 key comparisons we can do to assess whether this method may be any good. One is tumor versus met, so things that we know are clonally related. These are things that we want to be able to tell that they are clonally related by comparing CNVs, because the ground truth is that they are related. The other comparison is different patients' tumors. This is our standard for things that are definitely not clonally related because they're from different patients. And there's like 28 different possible pairwise comparisons that we could do from the tumors we had sequenced. And then tumor versus normal, we want to make sure that the normal background doesn't accidentally get called as clonal, so there's less chance of a false clonal call from lower tumor content. Okay, we tried a couple of approaches to this. The first one I'll tell you about is a manual assessment of eyeballing these plots and saying, by this criteria, do they look related enough that we can call these clonally related tumors? And for this manual assessment, we looked at 6 regions. The 3 Qs repeated because there's a couple different tumor genes in that area. These 6 different regions that are very commonly altered in lung plus other tumor types. And we just scored, is there an alteration of this region or not? And then does the potentially related tumor also have a similar looking alteration of the same region? So like within our resolution, you couldn't tell the difference in the start and end points. doing that for our, what's this, eight pairs of known clonally related tumors, they all had some number of alterations in the regions of interest between 1 and 4. And tumors that we know are clonally related had either half of those alterations in common or all of those alterations in common within those specific regions we looked at. We picked regions that have main driver mutations, thinking that those would be more likely to be clonal, early mutations inherited in the MET. So if you add up all those numbers, that works out to, depending on your criteria, if it's half, 50% overlap, then that picks up all of your clonal lesions, 100% sensitive, but not as specific. On the basis of these pairwise comparisons between different patients' tumors, some of those accidentally gets called as clonal, even though they're not, if you only need half of your regions in common. If you increase that to wanting a 100% match of these regions of interest, your specificity is better. There's no miscalls of unrelated tumors, but you only pick up about 3/4 of your clonal ones. It's not perfect, but you can kind of fine tune it for whether you want high sensitivity or specificity. And this approach will probably evolve too as we get more tumors to feed into our data sets. All right. I'll show you some examples of how these plots look. This is an example of a known primary MET pair. Even just eyeballing it, you can appreciate that there seems to be a fair amount of conservation in these regions. Here's another example of matched primary and MET. Again, there's a few differences, but overall, you're like, huh, this looks pretty similar. That gave me a lot of hope when I first got this data. And then if you look at different patients' tumors, they're really quite different. Comparing to a matched normal, all the normals are really quite flat until you get down to the smaller chromosomes. So there's probably a pretty low chance that normal is going to get miscalled as clonal. And indeed, in our analyses, there was no miscalls of the matched normal as clonally related. Okay, last approach I'll show you to this is, what if we tried to use math to do this more accurately? And this is drawing upon literature. Someone else published this very fancy-looking algorithm for trying to, in their study, compare whether DCIS and invasive breast cancer were clonally related or not, using copy numbers. So in this formula, it's more or less showing what proportion of the breakpoints of your CNVs are the same between your two tumors versus all of the breakpoints that you see in both of the tumors. It looks a bit fancier because they put a stronger weight for things that are shared than things that are unique, because the evolving clone could acquire unique things relatively easily. So they would weight those as less. In any case, this formula was applied. First, we made a reference distribution. And this is from the pairwise comparisons of all unrelated tumors from different people to see if you made a score by this algorithm and plotted it, how unrelated would they be? So most of these tumors had a score kind of down here. Very rarely the score crept up above 0.2. But we were able to make some thresholds that said, well, 99% of your comparisons between unrelated tumors produce scores on this side of things. So any scores on the other side of things probably predict that tumors are related. And maybe like 1% of the time you might accidentally call unrelated things related by that threshold. So if you apply that to our 8 known clonally related tumors again, most of them did score above the 99th percentile, so scoring as clonally related. There was 2 that did not, and 1 that was kind of in that in-between zone between the 95th and 99th percentile. So depending on whether you choose the 95 or the 99th as your threshold, you get sensitivities of like 63 to 75%, kind of on par with what we saw from the manual assessment. But this, again, I think we can refine further with a better histogram for setting this threshold. And the way to do that is with more data that we've been collaborating with the POG program and adding some of our own samples in as well. That hopefully in a future iteration of this, will have a threshold that might give even better accuracy values. Yeah, so as noted, future directions are to get more samples for this and refine these thresholds, maybe even making separate thresholds for lung cases than other squamous cell carcinoma cases, given the frequency of CNVs in these different tumor types are probably a bit different. Okay, so wrapping up then, this was a whirlwind through a couple different approaches, but it all comes back to these 3 key questions that were our challenges in practice. So in terms of that question of, if you have a squamous cell carcinoma, you don't know what the primary site is, what can you do? you could try methylation, and options seem to be arrays or nanopore sequencing, with different challenges and benefits to each, but both at least showing accuracy in our classifier, which if you ever have a methylation data set, you're welcome to try through the website link to the classifier. That question of if you have a patient who has squamous cell carcinoma in the lung and somewhere else in the body, and you're trying to figure out if they're related, like if, for instance, that lung thing is a metastasis or not from the other, you actually have two options then. You could try the methylation classifier approach to figure out the site of origin of your lung cancer, if it reads as a lung origin or if it reads as the other squamous cell carcinoma's origin. Or you could try doing the whole genome sequencing copy number assessment on both to see if there's evidence of clonal relationship in shared copy numbers between those tumors. So that is a bit dependent on whether you have samples of both tumors or if you have samples of just one tumor. You can choose which one. Finally, then, if both your tumors are in the lung, then you have to go with this CNV assessment because they'll all have methylation signatures of lung. But at least we have some preliminary data anyway that the shallow sequencing for copy number could, with moderate accuracy perhaps, give you some hints as to whether they're likely to be clonally related or not. All right, so I think my takeaway from this is that methylation profiling and shallow sequencing are assays that are, in my mind, clinically feasible. I mean, obviously none of these are like accredited or clinically validated assays yet, but I think just the way in which they work, the kind of data that they give, things like cost and input requirements are things that are compatible with what we think about for wanting to develop a clinical assay. And I think that the output of them in our classifier and in our CNV assessments is something that could be informative in the future for if you get a scenario that has two squamous cell carcinomas. So I know this is a very research-based talk. It doesn't help with the case on your desk today, but hopefully this is some food for thought as to ways we can approach assay development in the future to deal with these questions. And with that, huge thank yous to everyone who's been involved in this. BCHRI funded a big chunk of this, as well as the residency program. I'm happy to take any questions.
Thank you, Julia. That was a fantastic talk. So this is open for questions, and maybe I'll ask the first one, which you sort of alluded to in your very last slide, which is how do you see translating this into the clinic?
Exactly. I friends and collaborators are very precious, of course. And then a lot of it just comes down to having samples with a known clinical gold standard result to compare with. But one option might be if we were able to take some cases that have had focus panel done on them. Focus panel has copy number data on some of the genes that are covered by that panel. And then to see if we can find corresponding copy number alterations when we shallow sequence them. If we can do a set of those, that would be good data towards a clinical validation. And then, of course, all the political and personnel issues of getting it running in a lab is a separate thing. But I'm optimistic that if we can demonstrate utility for this, that everything else after that we can pursue.
Well, there are larger, the OncoPanel is larger, right? And would you be able to just add targets to that and use that as a proxy for whole genome sequencing if you're already doing an OncoPanel?
Yeah, it comes down to how much you need the breakpoint versus you need the driver gene. You can definitely get an assessment of whether the genes covered by that panel are copy number altered. But if they're common driver genes, they're probably going to be commonly altered in different tumors that might be unrelated. So I think if you have sequencing that can cover the edges of that gene in like the intergenic space where that CNV starts or ends, and that'll give you a lot more confidence as to whether something is truly a shared alteration or just by chance. But that said, I mean, certainly in adenocarcinomas, I think the gold standard for is a tumor related or not to another tumor is to look at, do they have EGFR mutations in common? Do they have ALK fusion in common? That kind of thing. And because for adenos, they already do focus panel, that's pretty easy to do. You just do one more focus panel. Whereas for Squame, they don't do any panel. So we have a bit more options to make a custom assay for these. And they have a lot more CNVs to choose from than an adeno would.
That's great, thanks.
Julia,
I'll throw it in.
Julia,
Question.
yeah, I wanted to ask you, almost all these squamous, for point number one there, almost all of the lung squams are in smokers, right? And I remember, Mike Stratton used to talk about mutational signatures and a very distinct mutational signature from cigarette smoking. We just talked about what you can extract from sequencing that's going on already. Is that just not practical because, your differentials are also smoke induced, or it's just, just wondering
Yeah,
how that tactic that I used to hear about would help for this issue.
yeah, for sure. If you're something like head and neck versus lung, probably they're both smoking associated and it won't help. But for other origins, that might actually be a good solution for a cutaneous squame versus somewhere inside the body, as if you try and find a UV signature. Could be doable. Absolutely. We do wish we had more skin samples in this study to try and loop those in. But absolutely. In fact, the simplest other option that I haven't really talked about might even just be looking at which p53 mutation is present in this tumor. That most of these squamous cell carcinomas will have a p53 mutation, and there's so many different mutations in that gene, it's probably going to be different if it's not clonal. Then you just need a panel that has p53, which Focus does not, but OncoPanel would, some of our local panels would, and then you just need a bunch more money to cover doing 2 panels. So there's definitely other options to this question.
Well, on a related note, could you use a smaller panel and cosmic SBS subtypes to classify these tumors?
Perhaps. That is also good food for thought. Yes, let me think further about that one.
There's a question in the chat. I'm not sure I understand it, but I'll read it out. It says, are we allowed to order Focus Panel or OncoPanel for diagnostic use only? And I think they're both diagnostic tests, but I'll leave that for you to answer.
There is some temptation that I sometimes feel of, if I don't know for sure if something is malignant, maybe I could just do a Focus Panel and see if it has a big cancer gene mutation, and then I would be pretty strongly swayed. That's probably a question for Stephen Yip, if CGL would approve you doing so. Historically, I don't think we've had a case where we've done so. It's consensus review. It seems to be superior in the local view, and I'm on board with that. But I think that our histologic impressions, I think I would trust more than an assay where maybe you're not sure what got into the core that went into the sequencer kind of question, or what tumor content that is. In terms of a Focus Panel, can you do it more than once per patient? That's another common question, which might be what you're getting at. It seems like with oncologist approval, it can often get by, like if it's going to affect how they manage this patient, per the oncologist, that CGL can be convinced, but of course they have the final say on things. And this issue of clonally related or not, I think in some cases I have seen them do a second panel to try and figure out if it's a recurrence or a separate primary, which has been informative.
Great, thank you. I think Stephen was here, but isn't anymore, so
No,
he
I'm
can't
here.
answer.
I can
Or-
answer.
Oh, there you go.
Yeah, we do make allowance for repeat testing, and particularly patients with multiple lesions in the lung. Is it like from the same driver? And certainly we have fine tumors with different drivers, and that dictates staging as well as treatment decision, which can be very different. With respect to squame testing, I think it's approved for squame non-smoker, but I think our testing criteria is really driven by clinical, financial reason rather than medical reasons. So even neuroendocrine tumor can have drivers as well, but we only have so much budget to test so much, and that's sort of it's just reality. With respect to testing like diagnostic sample that could be tumor or not tumor, Focus panel is great, but I think it's really lacking P53, and I think that's one of the key driver that like would be very useful to know about tumor or non-tumor. I recently have a brain tumor that I don't know was a brain lesion that don't know was tumor or not. I actually did a scroll and just sort of submit an entire scroll rather than a core because I don't know which area to core, and that might be one reason. But I think there are enough if there's sufficient clinical question that we think diagnostic will be useful, just email the lab or me, and then we can facilitate things.
Great, thank you. Are there any other questions? All right. Well, thank you very much, Julia. That was a great talk again, and looking forward to seeing progress.
Yeah, thank you. Thank you all for listening.
Thank you all for joining.